everybody. Welcome to join the webinar today. So we're gonna start the webinar from now. So today webinar is a co-production of our office, Israel Economic and Trade Office at the Embassy of Israel in Korea, and the Israel Innovation Authority with the support of KORIL-RDF Fund Office, the joint R&D fund between the Israel and Korean governments. So I'm gonna speak in Korean shortly to more explanation for the attendees. 안녕하세요. 저희 이스라엘 대사관 경제무역대표부 이민희 상무관입니다. 저희 오늘 웨비나를 시작하기 전에 간략하게 몇 가지 좀 말씀드릴 게 있는데요. 저희 오늘 그 제약기술과 바이오테크 혁신 기술을 가진 4개 회사가 참여해서 웨비나를 진행할 예정입니다. Biologics Design, Emendo Biotherapeutics, OnceCell Medical, 프레사이즈 이렇게 네 개 회사가 참여가 하게 됐고요. 저희가 이 웨비나는 저희가 녹화가 되고 있습니다. 추후에 필요하시면 저희 연락 주시면 저희가 이렇게 공유도 가능할 것 같습니다. 그리고 저희가 웨비나가 끝나면 B2B가 바로 있는데요. B2B를 신청하신 분들은 B2B 링크 받으신 곳으로 들어오셔서 B2B를 시작하시면 되고요. 그리고 웨비나 도중에 가운데 하단에 보시면 Q&A라고 있어요. 혹시 질문 있으시면 그쪽 Q&A 누르셔서 질문하셔도 되고, 그리고 추후에 아니면 저희 대사관으로 연락 주시면 저희가 질문에 대답을 해드리도록 하겠습니다. So, I'd like to give this floor Mr. Yaniv Goldberg, Head of Israel Economic and Trade Office in Korea, for his greeting.
Okay, so thank you, Minnie, and welcome everyone. My name is Yaniv Goldberg, Head of Economic Attaché of Israel in Korea. I will share my screen with you in order to go through my presentation. Today's webinar is a co-production with the Israel Innovation Authority and with the support of KORIL-RDF Fund from Korea. I'm representing the Israeli Ministry of Economy here in Korea. Second. So probably I don't need to explain a lot about Israel, but the fact that more than $8.3 billion in VC money went inside the Israeli startup ecosystem in 2019, and the fact that each year more than almost 1,000 new startups are being opened in Israel is something that is quite important to know. And indeed, as I was writing downstairs, Israel is small but truly disrupting technological paradigms all around the world. Israel also, it's not only me, it's also the World Economic Forum report that mentioned that Israel is an innovation hub where companies grow the fastest. And one more thing important to mention here, speaking also about R&D, is the fact that in Israel there are very fine academic institutions like the Technion and the Hebrew University in Jerusalem, but also the fact that Israel, together with Korea, are leading the world in terms of R&D, investment into R&D. Almost 4.5% of GDP is being invested into R&D. And this brings us to today's topic, actually. It's not only 4 Israeli innovative companies that we brought together, it's also some kind of a new concept that is being initiated by the Israel Innovation Authority, which is called Bio-Convergence, and it will be explained in details later on. But as I see it, Bio-Convergence is the sweet spot where the data and AI and engineering tools are meeting the pharma industries and biotech industries to create better manufacturing processes and better R&D. And having said that, I would like also to mention about the role of our office, the Israel Economic and Trade Office in Korea, part of the Israeli Embassy. What we do all year long, and especially in Corona times, is to bring the Korean companies together with the Israeli companies, either by providing scouting services for the Korean companies, but also trying to help the Israeli companies whenever they have a specific need in Korea. We also help Israeli companies to attract investments from Korean side. And obviously we arrange lots of events, not only this kind of webinar, but also actual events with delegations coming to Korea, delegations of Korean companies coming to Israel, events in Korea, but also across the world, with one purpose in mind: to create better cooperation between Israel and Korean companies. And obviously, Israel Innovation Authority is one of our major partners in Israel, and KORIL-RDF Fund is our biggest partner actually here in Korea. We work together very strongly. And I invite all of you to approach us after this webinar today, since we are the boots on the ground, both for the Israel Innovation Authority and both for the entire Israeli ecosystem here in Korea. So with that in mind, I would like to thank you all for joining us today. I would like also, again, to thank our friends and partners from Israel Innovation Authority and KORIL for co-producing this event. And good luck, everyone. And I would like to give the floor immediately to Mr. Tae-Hoon, Dr. Tae-Hoon Choi from KORIL for his talk. Please. Dr. Choi. You just need to unmute yourself.
Yeah, this is my turn. Okay. So you can see the screen. Boker Tov. Good morning to Israel, and good afternoon to Korea. I am Tae-Hoon Choi, the Chief Executive of Korea-Israel Industrial R&D Foundation, briefly KORIL-RDF. I am so glad to participate in organizing this meaningful webinar with MOEI and IIA, and to have a presentation of KORIL-RDF. Especially thanks to Mr. Yaniv Goldberg, representing the Israeli Embassy in Korea, and Ms. Ella Heller, representing the Israeli office of KORIL-RDF. Let me talk to Korean audiences for their understanding in Korean. 안녕하십니까? 저는 최태훈이고요. 한국-이스라엘 산업연구개발재단을 맡고 있습니다. 저희 재단의 영문명은 코리아 이스라엘 인더스트리얼 R&D 파운데이션이고요. 줄여서 코릴이라고 저희가 부릅니다. 그래서 영어로 코릴이라고 하면 저희 재단을 뜻하는 걸 이해해 주시고요. 오늘 양국 공동 세미나이기 때문에 제가 영어로 얘기를 합니다. 이해가 안 되시는 부분은 저희 재단을 접촉해 주시면 충실히 답변 드리겠습니다. Okay, my presentation is consisted of 5 sections and will be focusing on the KORIL program. By virtue of the similarity and difference between Korea and Israel, both countries are very complementary in terms of technology and industry. In this point of view, both countries signed on the agreement for bilateral industrial R&D cooperation in private sector in 1998. Based on this, both countries each contribute $2 million annually, and KORIL-RDF was established in 2001 in order to administrate, KORIL fund and support a joint R&D. At the ceremony last year, to declare the completion of the negotiation on FTA between Korea and Israel, both countries agreed with increasing KORIL fund up to $4 million per year from each country. Accordingly, Korean government and Israeli government started to discuss revising the agreement on KORIL program. So we are expecting that we can promote more numbers and better quality of joint R&Ds among the companies from both countries. And we are considering that Bio-Convergence would be one of the attractive industrial sectors where we can boost joint R&D between Korea and Israel further. KORIL's missions and objectives are to generate mutual benefits by promoting collaborative joint industrial R&D and to form an equal and collaborative platform for it. Furthermore, KORIL is aiming to encourage joint ventures leading to commercial success in global market and also to promote technology transfer between both countries. Basically, approved joint R&D projects are supported by KORIL with grants up to 50% of the total R&D cost to a maximum of $1 million. After the joint R&D project completed, 2.5% of commercial revenue will be repaid to KORIL as royalty. That will not be beyond the total funded grant amount. The companies in a KORIL project are liable for the rest of grant as matching fund at least 50% among the total budget. KORIL program opens to all the industrial sector except defense industry. The project will be comprised of at least 2 companies that must be registered and operate in Korea and Israel respectively. Subsidiaries of companies headquartered outside Korea and Israel can apply KORIL program when they satisfy the conditions as below. There are 3 types of projects in KORIL program as for now, such as feasibility study project, mini-scale project, and full-scale project. In a feasibility study project, you can design a joint R&D project investigating technical feasibility, market acceptability, related patent survey, and so on for 3 months with $30,000 of grant money. In a mini-scale project and a full-scale project, you can conduct a joint R&D. They are classified by project term and amount of budget like this. In principle, stipulated in the agreement for KORIL program, the grant cannot be more than 50% of total budget, as I mentioned in the previous slide. The project term of a full-scale project would be from 1 year to 3 years, and maximum of the grant money will be $1 million like this. This slide shows the procedure of KORIL program. As for a full-scale project only, the final decision whether to approve it will be made at the KORIL board members meeting. Usually, it'll take 5 or 6 months from RFP to the final approval. Up to now, among 315 proposals, KORIL approved 176 projects with $68 million of grant money through 37 funding rounds. Approximately 70% of the approved KORIL projects were from ICT and electronic industrial sectors. Total 38 companies reported sales outcome throughout 75 KORIL joint R&D projects. As for now, the total sales outcome is about $36.5 million. For your partner matchmaking and consulting, please refer to this information of the Korean office and the Israeli office. And also, for your more understanding and faster information, I recommend you that you visit in this. Thank you.
Thank you, Dr. Choi, for your words. Now, I would like to give the floor to Ms. Anya Eldan, Vice President of the Israel Innovation Authority and Head of Startup Division.
Good morning. For us, it's morning. I was very lucky to visit Korea, what was it, about 6-7 months ago, and meet a lot of people and talk about bioconvergence. And it is very exciting for me that such a short time after we're having this seminar and trying together to build the bioconvergence industry. So I will not take a lot of the time because I think it's more interesting to listen to the companies. But I would like to give very short overview once I handle the problems. Okay. So Israel Innovation Authority is very shortly a government agency. Our budget is about $500 million US. About 1/3 of the budget goes to life sciences investment, and that includes pharma, digital health, and medical devices. And we have 3 roles. One role is to strengthen the innovation ecosystem. As everybody said before, Israel has a very strong innovation ecosystem, very similar to Korea in a way that both our countries invest a lot in R&D, twice more than the average of OECD countries. And one of our jobs is to strengthen that and do everything to make the Israeli industry competitive. The second role is to enhance the economic impact of high-tech industry. And that usually means when multinationals come to Israel, we try to get them to bring a lot of value to the industry. So not just scouting for technologies, but actually working with the companies here. When we look at companies that we support, we'd like them to be complete companies, so they don't just develop technology and sell it outside of the country, but actually develop most of the product in Israel and hopefully manufacture. And our third role, and this is what I'm going to talk about today, is to help create future growth engines. So emerging disruptive technologies, those are the ones that create the value, the economic growth, and our role is to try and identify those and to promote them. And bioconvergence was selected last year And it's a name that we coined, but it's really a combination or integration between biology and life science and other engineering disciplines. So you see here physics, materials, nanotechnology, electric engineering, and a lot of AI, of course, and big data. And the convergence or transintegration that occurs when biology is combined with those disciplines is what we call bioconvergence. And the reason we're excited about this is that we think that's where the healthcare is going. So I'm putting some examples. I'm not going to read all of them. I'm just going to talk through why we think this is a future growth engine for Israel and why we think it's a future growth engine maybe also for Korea. And that's why we want to join forces, because we are very complementary economies in those ways. So the future healthcare is going to have to change. We all understand, I'm not going to go why the pharmaceutical industry is going to have to change. The economics just have to change at some place. And the new technologies, the interesting thing about bioconvergence is that it wasn't born last year. The academy did academic research for the last 30 years. So a lot of the examples you see here are results of very deep and deep research, and sometimes I like to think of it as really science fiction. So when you think of some of the companies mentioned here, probably when they started, somebody said, this is never going to happen. But after many years of research, some of those companies are already coming close to the market. So NanoRetina is one example of a company that's creating an electronic retina. They already did first in man. They actually did 2 or 3 or 4 companies, 4 humans, and with successful results so far. So I think the science fiction is becoming a reality. And our decision as a government agency was to do what we can to help this tech transfer. Bioconvergence is one area where a lot of IP is created by the universities. So this is a very complex, multidisciplinary area, and a lot of the innovation comes out of universities. So our role is to help the tech transfer and help bring together different partners who can join in this transdisciplinary research. I say transdisciplinary because this is not engineering product and a separate biology product, and then you sort of combine them into a joint product. No, this is a research that is very deeply interrelated. So the biologists have to understand the engineering, and engineers have to understand the biologists. And maybe in the future, we will see this sector led by people who studied both engineering and medicine and biology, and actually understand all of those disciplines and how they integrate together. So there are a lot of challenges, but things are beginning to happen. So I have here a couple of examples. Wyss Institute in Harvard is one of the leaders in this type of transdisciplinary research and have already created over 20 companies that came out of that translational research. Galvani was established by a pharma company with engineering company, which is not that many examples that we see yet. And I think this is a challenge for us, how to create this synergy. And the government policy, not just Israeli government that decided to take this approach, but you can see here also NIH is increasing the coverage that they do in convergence research. And we think Israel is very well suited to be one of the leaders in this strategy. First of all, because, and mostly because, we have a very strong multidisciplinary capabilities. A lot of the big inventions in the Israeli market, Israeli success, are very multidisciplinary products that combine different disciplines, and we seem to be good in this area. And the government has already invested quite a lot of money in different initiatives that are contributing to bioconvergence, like the Digital Health Initiative and the Nanotechnology Initiative. So we plan, this is one of the reasons we decided to invest in this space. We're creating a lot of different initiatives, from encouraging multidisciplinary research with dedicated grants to special translation initiatives that help transfer this advanced research into industry through different schemes. And the reason we're talking here is the last on the list, how to attract international collaborations. We don't believe the industry is going to change just by Israeli companies. We believe it's going to change by collaborations. And we thought Korea and Israel would be ideal partners because we have a very similar approach both to research and to R&D. And my last visit there, which all the people here helped organize, was very, very interesting. And at least one collaboration happened as a result of this visit. So I know that it's possible. I met one company during my visit, and I introduced them to another company in Israel, and they're working together. So I'm very hopeful that as a result of this webinar and the good work that Dr. Choi is doing and the team, we will have more such collaborations, both between Israeli industry and Korean research and academic institutions, and especially between Israeli companies and Korean companies. So with this, I wish you luck for this webinar and the collaboration that I hope will result out of this webinar, and thank you all so much.
Thank you very much, Ms. Eldan, for your interesting talk. So now, I would like to give the floor to Biologics Design. Please.
Great, so thank you so much for the time and invitation. Thanks to the Innovation Authority and KORIL-RDF and to the Economic Trade Office in Korea. My name is Micah Pearlman. I'm Chief Operating Officer here at Biologics Design. With me today is Ava, who's the BD Manager here at Biologics. And over the next 10 minutes, I'll try to illustrate a little bit about what we do here at Biologics Design. So, as you can see, what we say about ourselves is that we try to unlock novel therapies using our computationally designed antibodies. So basically what we mean by that, and I'll illustrate that in a minute, is not that we use AI to do the same kind of technology used today for drug discovery and drug development better, faster, cheaper, which is a common use for AI technology. What we're really trying to do is use specific algorithms to design therapies that otherwise wouldn't have been possible. And to give you a sense of what we mean by that and what we're trying to solve, if you look on the left, basically in drug discovery and drug development, what we're trying to do is create a molecule that will kind of be thrown into the biological network that we see there on the left and create a profound effect that will really alleviate patients' illnesses and solve many problems. Now, we are very constrained today. Most technologies for drug discovery and development are very constrained on their ability to do that. And many of the reasons really come down to the fact that what we're trying to do is find a small molecule or an antibody, basically, or a biologic that will get into that biological network, find a target as depicted on the right, in the zoom in you can see on the right, and try to hit on that specific target a meaningful place that will really have a profound biological effect. And we're doing that using technologies that have many limitations and are rather statistical by nature. So you're not necessarily hitting the exact right place you need to hit to get that effect. And usually you're doing this in the context of only one kind of effect at a time. You're affecting one target at a time, even though the biological network, as we see on the left, is very complex. So at Biologics Design, what we try to do is what we call clever antibodies. We have our AI capabilities, enabling us to create antibodies that are normal IgG antibodies, and they're not any non-biological constructs. They're just like any antibody your body would make on its own, but it's designed and programmed to do very amazing things. So for instance, our antibodies that we call multibodies can bind more than one target in different contexts. So we can actually program an antibody to bind more than one target and in different conditions. So for instance, we could create an antibody that will bind one target in the presence of one tissue and another target in the presence or lack thereof of different tissues or different other types of molecules that are around it, or in different pH contexts, etc. So you could really think about it as a programmable antibody. Another thing that's very interesting that we could do is use our capabilities to design antibodies that will bind targets that in others, the normal technologies for drug discovery are just undruggable. We can't, and nobody has been able to create antibodies for those specific targets. One example that I showed actually in the previous slide was that ion channel, which today we don't know of any other company that actually designed an antibody that would block successfully an ion channel. And using our AI capabilities, we were able to do that very quickly. So this is how we go about unlocking the new novel therapies. And how do we do it? First of all, we go and we start thinking of biology in a more creative manner. We go to the biggest KOLs or pharma companies, the biggest experts in a specific disease domain, and say, let's go crazy. Let's think about the actual biological context we want to solve. And let's not limit ourselves to the limitations of current drug discovery technologies. Let's just decide what we need to do and see how we can do it. Then we apply our algorithms, which I'll give a sense to in a second, to design the exact type of molecule we need. And once we have the design, the design that's based on our computational approaches, we go out, we synthesize the antibody, and then take it through the normal path of drug development. So basically what our AI enables us to do is to create that first molecule that would do the incredible. And then from that point on, we would take it through the regular development path of any drug, in vivo, in vitro, and obviously phase 1, 2, 3, etc. So just another glimpse of what do I mean by a multibody, right? So I said our antibodies are regular, normal IgG antibodies. And they're not like any of these bispecific or other biological constructs. They're just normal antibodies. But if we look on the left, you could see that a regular antibody here, depicted in gray, would bind one target at one time or place. In this case, a square. What our multibody can do is have its arms compatible in this illustrative case with both a square and a triangle. Okay, so these are two completely different targets in different tissues perhaps, or in different contexts, but the antibody can bind either or, and so basically now you have one antibody that can do three things versus one. It could bind either the square, it could bind the triangle, or it could bind both the square and the triangle simultaneously. And again, like I said, we could program this to be done in a fashion that would be in the an if or kind of context. So it would be a program type of approach and not just statistical. The secret sauce behind this is obviously our machine learning capabilities. I won't delve into this now, but basically what we do is we have a set of algorithms that mimic the way the body creates an antibody. We start by understanding what target we need to design the molecule to bind, or in what context. We have then a database of template antibodies we start out with. Our machine learning algorithms pick a template antibody, and then we have another set of algorithms that help us decide which changes we would have to make to that template antibody in order to get it to bind or do the exact program that we want it to do. Once we have that, we have the design, we go out and synthesize a library. It's a very focused library of antibodies. We test it here in our lab. We see, if it does exactly what we want to do or any improvements need to be made. And then we have an iterative process of 1 or 2, 3 cycles in which we improve and finalize the final antibody. So the culmination of all this basically leads up to this unique pipeline of antibodies we've been working on for the past couple of years. and I won't go through all of them. I'll just mention that each and every one of them has a very specific uniqueness about it. So even if we take our lead asset for oncology, which is for a very well-known pathway and target, the way we're going about it is also just enabled by our machine learning capabilities. What we're trying to do, what we're actually would successfully have done, we have lots of data showing it, is we're one of the only companies, we're actually the only company that has shown that we're able to direct an antibody to the IL-2 cytokine and blocking a very, very specific path of that cytokine. No one else has been able to do it so surgically. And most companies have just avoided trying to create antibodies for this specific cytokine for many reasons for many years. So this is a potential best-in-class type of asset. We have other assets in the pipeline around autoimmune. And if you look down at the bottom of the list, BD6 and BD7 are our first multibodies in the pipeline. And these are antibodies, again, that are normal standard antibodies that bind both PD-1 and GITR in the case of BD7, or PD-1 and OX40 in the case of BD6. And we have also in vitro data showing that they're efficacious. Great. So just a little bit about who we are and the team. So this is just a little bit about the leadership of biologics. So you're meeting me today. This was founded by Professor Yaniv, who created this technology and is the CEO and founder till this day. We have Tim Wines, who's the head of early development. He's situated in Boston, and he spent over 20 years in Takeda and Millennium Pharmaceuticals. Has taken many antibodies from design and discovery all the way through approval. And he's in charge of taking all the antibodies that we design and discover here in Israel and start taking them down the path towards phase 1, 2, and 3. We have also some significant advisors, starting with Michael Hayden, was the CSO of Teva. Werner Meyer headed biologics at Biogen. John Haswell was the head of all biologics in Novartis, just recently retired and joined our board, our science advisory board, and Shmuel Kabili. We have 27 scientists here in Israel, 12 with PhDs. We're situated in Rehovot, the Science Park, and as I said, we also have an office in Boston, which is headed by Tim. And that's it. And in terms of what we're looking to do, we're now raising some funds to advance some of our early assets through preclinical proof of concept, and then from there on, we will take them through IND enabling studies and into the clinic, and also enhance our AI platform. I will say that out of this round, we already have a significant commitment from a big pharma company we're working with. So we're really in the, I think, pretty last stages of fundraising. And other collaboration goals we're looking for with pharma companies, potential pharma partners, is either partnering on some of our pipeline assets or collaborating with any one of you out there in the audience that would like to create a new therapy or have targets you're looking to affect. We could definitely leverage our capabilities to do that. We've done that many times with many pharma partners, some of the top 10 and top 20, and are looking for more innovative and fun projects. So I'll end at that and give a minute or two for Q&A.
Thank you, Micah. So now, I would like to give the floor to Emendo Biotherapeutics. Yes. Emendo, you can start.
Can you share the presentation?
Yes.
So while we're putting on the presentation, I would first like to thank all the organizers from both Israel and Korea for giving us this stage and opportunity to present Emendo and meet potential collaborators from Korea. So thank you all for that. I'll just give you a very, hopefully short enough introduction and tell you a little bit about Emendo. First, Emendo is a next generation CRISPR company. I'm sure that most of you, for sure, the ones that come from the biospace know CRISPR. So what is next generation CRISPR and what is it needed for? So when we established the company, and it was 4.5 years ago, we were looking at the potential challenges or bottlenecks that...
Sorry to interrupt for a second.
Sorry?
I'm sorry to interrupt, but your screen is not shared. Is it me?
Oh.
Hold on, we'll stop and share again.
So please reshare it.
Yeah, we're doing that.
And try to be on time, please. Thanks.
Are you able to see now? Are you able to view our screen?
Yeah, we
Yes.
can see. Thank you.
Okay. So Julie, you should follow up on time.
Yes, sure.
Okay. So next generation CRISPR. So again, when the company was founded 4.5 years ago, we were trying to look ahead and identify the challenges that this exciting technology will face in the future, in order to bring it to allow it to provide as many solutions as possible within the medical space. So the first thing that the first problem that we wanted to tackle was the precision tackle. We wanted to practically get the highest precision possible with gene editing. I don't have to tell you why getting the highest precision possible is essential. It's a safety issue. This is, it's super exciting that we are able to actually manipulate the genomic DNA, but it brings with it a lot of risks. You want to cut only where you have to cut, and you don't want to cut anywhere else. So getting the highest precision is the first thing, safety related, that we wanted to tackle, and that's what we did. When we were able to get to very high levels of specificity, practically the highest levels possible, and in many cases eliminating off-target altogether, we decided to take the challenge of allele specific editing, and I'll show you, I'll tell you more about that later. But allele specific editing practically means that you want to genetically manipulate or edit a single allele while leaving the other allele intact without cutting it at all. When you're able to do that, it opens the window for a lot of therapeutics that are not available otherwise. Another thing that we decided to do, and that's the Omni technology. Very early, we realized that with all the complexity in the CRISPR space, we want to identify additional nucleases, CRISPR nucleases, that will, one, potentially have freedom to operate within this complex space. And in addition to that, will have different characteristics that, again, will allow to target organs and genes that otherwise cannot be targeted. Just a very short company overview. So in Emendo, we have around 60 employees. Most of them are based here at the Science Park next to Weizmann Institute of Science. We're also located and headquartered from the US, and a lot of our collaborators and the programs that we actually developed are in collaboration with different institutions in the US. In terms of technology, as I said, novel CRISPR nucleases, that's the main focus for Emendo, allele specific editing, elimination of off targets. I've mentioned all of that. We also design proprietary editing strategies. There are many ways to actually manipulate a gene and get the effect that you want. You can knock out, you can do HDR and correct. You can even take whole parts of a gene and insert other segments instead of it. So that's also a significant focus for us. We do gene editing in various cell types. That's also important. You know, this very powerful tool, that we call CRISPR. At the beginning, when we started working with it, we thought it works like magic. You just throw it in a cell and it works. Well, that's not the case. It doesn't work in a large portion of the cases, and not in every cell. And these are some of the things that we have solved with our platform in Emendo, being able to target any gene in any type of cell and any site within a gene. We're very IP focused as everyone in this space, 36 patent families altogether covering the technology, the platform, and some of the indications that we target. In terms of business, we raised over $70 million to date. OrbiMed is our lead investors. For those of you who know, it's a significant US and Israel-based fund. Also, Takeda Ventures is a significant investor in Emendo. And Aegis, which is a Japanese biotech company, also a new partner to Emendo, and we work in close relations with them. So Emendo, being able to provide a solution within the gene editing space is a complex thing. It's a complex task. Actually, when I look at the first indication that we're targeting, I think it's one of the most complex medical solutions that there are. It's an ex vivo approach that requires a lot of stages along the way in order to bring it and make it actually valid clinically. You need to be able to produce it, and you need a lot of expertise along the way. So our discovery stage starts with our computational team. Then we have the experimental side that actually continues discovering the new CRISPR nucleases. Then we have the protein engineering team. Our protein engineering platform, generally, protein engineering is a significant focus for Emendo, and we have created a whole platform that is very, very specific to engineering of nucleases generally, but more specifically CRISPR nucleases. Guide design, we need to target it to the right place, and this is also a challenge. Then we have proof of concept essays and a development team. And then we reached the development stage itself. As I said, all of our programs are being developed together with partners that I'll tell you about some of them later on.
1 or 2 minutes.
Okay. Why allele-specific editing? Just what you see is marked in orange. These are the diseases that will benefit from applying allele-specific editing. So actually, if you're not able to do allele-specific editing, you're left with a relatively small portion of the genetic disorders that are actually available to you. So we are focusing on these diseases, which are actually, around 80% of the old genetic disorders. This is just a summary for our optimization platform. Won't get too much into that. I'll just say one thing. We're covering all types of cells along this process. We start in high throughput bacterial cells, and we finish the process by screening in the target cells themselves. For example, if we want to target, for instance, one of our interests is within the CAR T space. So part of the screening or engineering process will go through screening in T cells themselves. And this proved itself to be very, very efficient. We target various targets, and again, this is a platform, so we have a lot of programs going on. We're starting with hematology, severe congenital neutropenia. This is the first indications that we are taking towards the clinic. It's being developed together with the University of Washington and Seattle Children's Hospital. We have additional indications that we're developing in hematology. Following hematology, we're working also on ophthalmology together with a collaborator from Columbia University, dermatology, oncology. These are all targets that we're actually developing these days.
Okay, David, sorry to interrupt again, but let's wrap it up in order for the other companies to have some time. Thanks.
Well, that's the summary, but we've covered everything. So thank you all for listening, and I'm wrapping it up. Thank you, guys.
So thank you, David. So now we would like to give the floor to OnceCell Medical.
Okay. Hi, everyone. One second, let me see that you're working. Yes. Hello, everyone. My name is Yaron, the CEO of OnceCell Medical. I would like to start by thanking the organizers for this fabulous event to present what we're doing. OnceCell Medical is a personalized oncoimmunology TCR-based therapy using our discovery device. I'll start with the team. The company was founded by Professor Uri Sivan, who's the current president of Technion, and also by Dr. Elad Brod, who's our CTO. And we also have in our team Dr. Shachar Ben Shaul, who's our senior scientist. I'll jump straight into the need in the case of cancer cell therapy. As you all know, there are two approaches being used today in cancer therapy. And the first one is the TIL, the tumor infiltrating lymphocyte therapy, basically taking the lymphocytes from the tumor, isolating them, enriching them, and then putting them back into the patient. And this is working today only in melanoma. It's limited to melanoma. And it has another disadvantage, although a good therapy in the case of melanoma, is the fact that nobody really knows what they're putting back into the patient in terms of understanding what are these specific receptors that identified the tumor or the antigen within the tumor. So this is one approach. The other approach, already known to everyone as well, the CAR T therapy, basically taking the lymphocytes from the patient blood, engineering them with the CAR T element, and then reintroducing them, infusing them back into the patient. And this therapy has good results, but it is a monotherapy, which is one issue. And the other one is that today it is limited to non-solid tumors. And basically, if I had to think of a desired therapy, I would look into a therapy that will not be limited to anything, meaning today, basically to solid, not limited to non-solid tumors, but rather available also to solid tumors, which is 90% of the cancers. In addition to that, it would attack more than one target, meaning not just one epitope or antigen within the tumor, but rather several. We would like to know what these target identities are. And in our perception, this can be achieved by a personalized approach rather than a generalized one or shared antigen one. Altogether, it will provide a low resistant potential. And this is exactly what our discovery device solves. So how do we do it? Here you can see, and you can follow my mouse, you can see we have in our device two arrays. This is the cage array, and basically you can see here a cut through the array. So you can see we have the wells themselves, and we're using a very gentle pumping of the cells into the wells. Now the thing is that once a cell gets stuck within the hole at the top of the array, while the solution can get through, but the cells cannot, in that point, the pumping will stop in this well, and it will enable or direct or redirect the other cells into other wells, which provide us a very high single cell capture efficiency. And you can also see it in live. This is exactly what we see under the microscope. That is one of the biggest advantages in our system, is that we can literally see the cells. You can see here there's a cell and it's stuck within the cage with one of the wells. So how do we use it? Once we have the cells being caught within each one, within its own well, We do specific anti-tumor T cells detection. We're basically using immunofluorescence analysis. It's based on regular methods. We're not inventing them. And then once we do that, we add the second array or the second part of our device. We call it the DNA array. Basically, it's a slide that seals the first array, the cage array. And by doing so, now we have in each well one cell that is sealed within. Now, once we do that, we can actually analyze the T cell receptors of the mRNAs and the mRNA of the T cell receptors, sorry, and then sequence them. In altogether, I would say that what we're doing is that we take the T cells from the, we can take the T cells, we can actually capture them and characterize them. And once we have the information that we're looking for, meaning the T cell is specific against the cancer, we actually can retrieve the sequence of the anti-antigen, the specific antigen, anti-antigen receptor of these cells. The thing is that we're not inventing the wheel. We're not trying to do something that is all new. What we're trying to do is to harness existing know-how and create maximum value. And we do it using current know-how of therapies. So this is the streamline of what we are doing. Basically, you have the biopsy taken from the patient as it is being done in TIL therapy, tumor-infiltrated lymphocytes. Instead of taking the T cell and then after isolating them, starting to enrich them, we actually take them into our device. In our device, we can identify the cancer-specific T cells using specific antibodies. And once we do that, we seal the wells. Each one has its own single cell caught in, and then we can sequence the T cell receptors and identify the ones that are actually from the cancer-specific T cell we just identified. Once we have these sequences, we're actually jumping back into already known art, meaning using the same concept of CAR T therapy, basically taking new lymphocytes from the same patient, engineer the sequences into these T cells, and then reinfuse them back into the same patient. We believe that our solution using single cell analysis as a personalized anti-tumor T cell receptor sequence enabler, it will provide a rapid and information-based, a precise and robust tumor adjustable T cell therapy. And I will explain soon what it means, why is the adjustable is so important in our perception. Our work plan is to finalize the optimization of our system within the next 2 months. We expect to reach above 85% of the RNA sequences from the cells within our system. It will be followed by a first proof of concept melanoma clinical samples, and the second half a year will be for studying additional clinical samples right now in head and neck and breast tumors. This is also aligned with our business plan. We think that once we're going to reach the first proof of concept in melanoma clinical samples, it will provide initial interest already with investment entities. And we do believe that by the end of the year, once we have the results from additional tumor types, it already will be of high interest for potential strategic partners. I have to say that we do understand we are a small company, we're a very early stage company, and we are seeking strategic collaboration constantly because we do understand that this is our best way to expedite the time to market. In terms of status, we are a third-year incubator program company in the Israel Innovation Authority. The company already had investments of $1.4 million in the company by the Israel Innovation Authority, by Sanar Ventures, and by the Technion. And we are seeking for $1 million in order to expand the team, to continue R&D, to finalize the proof of concept study in clinical samples in melanoma, and also start our solid tumor study in head and neck and breast tumors. Also, we are going to establish a strategic fast-track regulatory track in clinical trial initiation, and at the same time, or in parallel, we are working on approaching global partners in order to facilitate good collaboration. I would just like to say one last thing, and that is about the adjustable point. So the reason that we think that the personalized cancer cell therapy is so important, because imagine that you have a patient that comes back to the clinic with the resistance to the treatment. Now, what we are doing is basically just harness the information that already exists within the immune system, which is a very flexible one. Every time the tumor changes, the immune system already creates the new solution also for that tumor. So once such a patient comes back to the clinic, what we're saying is that using the same exact approach, we can actually create new T-cell receptors that will enable us to retreat the person. So basically, our vision is not just to create the first-in-line treatment, but actually provide an ongoing treatment. And by doing so, we hope and we believe that this will create a new concept of cancer therapy rather than a lethal disease to make it a chronic disease. And this already has been done in HIV, not exactly like we are doing it, but the concept already is there. I just want to thank you everyone again, and I'm open for questions.
Thank you, Yaron, for interesting presentations. Now, I would like to give the floor to Precise.
Precise Bio. אריה, בבקשה.
Okay. Good morning in Israel, good afternoon in Korea. Thank you for the opportunity. Precise Bio is a company in the field of tissue engineering, biofabrication of tissues. And when we talk about bioconvergence, I think that we bring the whole scope of the technologies from the biology, biomaterials, engineering, 3D printing, and at the end also the preclinical and clinical studies, bringing all of this together to fabricate tissues in the same anatomical structure as they are in the body. And this is done inside the company from the cells through the printing to the maturing of the tissues and the transplants of tissue, the way the tissues are structured in nature. The company, at a glance, started out from a unique 3D bioprinting technology where we can deposit cell by cell as the structure is in the body. We have validated tissue engineering processes. It's not just that you have a cell, but you have other materials that you have to bring them together in order to fabricate a tissue. And we have a platform essentially that can enable us to bring these kind of tissues to the market, to the clinic, and not just to do it in a lab, but really scale it up. Our 3D bioprinting technology, which is a proprietary technology based on a laser printing technology, really can enable scaling up these tissues. So it's not just doing it one time in the lab, but really to bring it to the clinics, bring it to the clinicians that can bring these tissues to the patients. We have started out in the ophthalmology field as a first area where we have transplanted a 3D biofabricated cornea in preclinical studies, and it's functioning. The company itself is led by world leaders in the field of tissue engineering who have been working in the field for the last 20 years, and we look at it, bringing all of these together. Really, there's a multi-billion-dollar pipeline of unmet therapeutic needs in various areas. As I mentioned, we started out in ophthalmology with the cornea transplant, but the technology enables to move forward into other areas also. So as you can see just at a glance, this is really a company that brings bioconvergence from the technology, from the science, through the technology, through the engineering, and out to the market. Over the last 20 years, many of these technologies have been evolving. They started out with basic science, and in the last 20 years, if we take, for example, starting with any tissue starts from the cells. So if 20 years ago you would be able to expand 3 types of cells from the body, now maybe there are 3 types of cells that cannot be expanded. So these sciences have been evolving over the years, and essentially there's a process that has been proven in the lab. And I'm just showing here one example of a urethra where you can take a biopsy of cells from the patient himself, extract the cells from the biopsy, culture them, expand them until you have enough cells to structure a new organ, and then structured it in the lab. And then you can have a new organ that is fabricated from the patient's own cells. And I'm showing here on the right side at the bottom, somebody that was injured in a car accident, and as a result of the trauma, his urethra was damaged, and he received a fabricated tissue from his own cells, and with 6 and 8 years follow-up, essentially he has his own organ, a new organ that functions in the body, fully functional. There's no rejection, no rejection from the body because it's fabricated from the patient's own cells. And this was proven in several types of tissues. So the science over the last 20 years has evolved and developed to enable these kind of processes. But as you all know, we don't see these things in the clinic today. And really the reason is because the fact that you can do it one time in the lab or repeat it several times in the lab is not enough to get it through the regulation. It's not enough to bring it in a sustainable, manufacturable way to the clinic where clinicians can bring these solutions to the patient. And here is where Precise Bio comes in. And we are combining our unique 3D bioprinting technology and taking these sciences and bringing them, I would say, making an engineering solution that can bring these sciences and clinical solutions to the market where you can produce large quantities and you can do it reproducible. Each time you're going to get the same cell density, the same structure. It's a quality controlled process where you know what you're going to get each time. You can measure it, you can control the process, the thickness, the mechanical properties of the tissue. And of course, this is also very cost effectiveness because you do it with a manufacturing tool. And this is what Precise essentially does. It takes existing science and with engineering concepts, really bring these technologies to the clinic. As I said, Precise started out as a platform, and in the past we have printed hepatocytes, liver cells, cardiomyocytes. We have looked into the field of orthopedics in skin and kidney in the future, but now we're really focused in the ophthalmology field. That's the first field that we are active in. All the other we will be approaching down the road, but as the first area of interest, we're focusing in ophthalmology. Why ophthalmology? And let's think, for example, of a cornea. If you have a damaged cornea, a diseased cornea, you will be losing your eyesight. And here, when we put in a biofabricated or 4D biofabricated cornea, the success criteria is very clear. The patient sees or he doesn't see. It's not, for example, if we biofabricate a part of a tissue of the heart and you do a follow-up of 5 years, you won't know if the patient had not had another heart attack because of statistics or because the patch has really done something. So here with ophthalmology and the cornea specifically, it's very clear of the outcome, clear and immediate result. The regulation path here is relatively clear and straight. We are probably the first company that's going through this process, as the FDA stated, but it's a relatively simple process. And the field of ophthalmology in general is a field that can adopt innovation. Other fields in healthcare are a bit more conservative. And another additional thing, and when we think of the cornea as part of our eyesight, where if somebody has an aberration or a default in the cornea, so we can also improve the vision. And of course, we're approaching here multi-billion-dollar markets. I'll just show a few.
Sorry to interrupt for a second. First of all, we have a question in the Q&A section. If you can see, you have a question for Precise Bio. This is one thing. If you can answer it shortly live, it will be great. And then I would like to ask you to wrap it up because we need to move all of us to the B2B section. So try to wrap up your presentation with that Q&A. Thanks.
We have not done yet clinical studies in general. We have done, we have printed a skin, but we are not focusing on that at this stage. We are focusing in the ophthalmology field. I'll just show results, a brief results of what we have done. We see here at the top row corneas that have received our transplantation. 4D biofabricated functional tissue, which functions in rabbits. You can see what the disease looks before without our tissue, where the cornea is opaque. I mentioned that we can change the curvature of the cornea, and here you can see a transplant that we have done also inside, inlay inside an animal's eye, where essentially it shows that we can improve the vision in this process. The founders of the company are myself. I come from a background of physics, and my two partners are the leaders probably in the field in the world. Dr. Anthony Atala from Wake Forest, who was the first surgeon to transplant an engineered organ over almost 20 years ago, and Professor Shai Soker, a professor of molecular biology at Wake Forest, previously at Harvard. We are seeking partnerships in various areas, also in the R&D in the field of lasers, platforms, biomaterials, and cell technologies. And moving forward also to the clinic, we are looking for collaborations. And of course, local distribution in the East is something that we are seeking.
Okay.
So thank you for the opportunity. As I said, we're really a company that combines inside bioconvergence from end to end, from the cells through the engineering, all the way to the clinic. Thank you.
Thank you, Aryeh. Thank you very much for the presentation today. So we'll move to the B2B session now. So from 4:10, we're gonna start the B2B. So someone who registered B2B today, please go to that link. And please keep in mind the B2B meeting is for 20 minutes. So we have every 20 minutes, we have another meeting. So thank you very much again.
Okay, and I would like to thank on behalf of all of us, the Israel Innovation Authority, our office, the Israel Economic and Trade Office, also KORIL-RDF, the Joint Fund. Obviously, thank you to the Israeli companies and all the Korean participants. I would like to mention again that if you would like, if you haven't registered for a B2B session with the Israeli companies, we can arrange some more meetings in the next couple of days. So if you're a Korean company participating today, you can write us an email and ask us to arrange you a meeting, and we will do our best. I would like to thank you all. Have a pleasant day, and let's move for fruitful B2B meetings. Thanks you.
Thanks very much.